Okay, awesome.
Yeah, very good. Thanks.
Great. Okay. All right. Well, why don't we get started? Welcome everybody to today's symposium, the general purpose of this series is to try to kind of explore what are the kind of major open questions in human neuroimaging research, and also kind of from a methodological bent, what is the future? What should we do as human neuroscience researchers in terms of the new methods, the new approaches, new techniques that we should adopt over the next decade. And it's kind of, it emerged out of a need that we perceive at Caltech in the human neuroscience community, that we want to kind of start to think about what the next level technologies are and next directions that we're going to go in ourselves in the next 5 to 10 years. So that's why we've invited speakers who are experts in their relative fields to try and kind of get a sense, a survey of what's going on and what the future is. And today, Tom Liu who's from UCSD, who's a professor of radiology, psychiatry, and bioengineering at UCSD and director of the Center for Functional MRI, is going to talk to us about multimodal imaging for the purpose of characterizing resting state global brain activity. Without any further ado, I'm going to pass over to Tom, and thanks very much.
Great. Thanks a lot, John, and thanks to the Caltech Imaging Center for this kind invitation. So yeah, I'm going to talk a little bit about the work I've done and many others over the past decade or so, using multimodal imaging to look at resting state global brain activity. I'm going to start off talking a little bit about some of the controversies and why it's of interest, and then some of the more later work looking at what might be driving this activity and some of the discoveries that are going on. Okay, so just so everyone is on the same page, as most of you know, in resting state fMRI, which is the place we'll start, is we're really looking at spontaneous fluctuations of brain activity. So if we look at any given voxel, it looks quite random, as the subject is lying in the scanner, typically with their eyes closed or maybe on a fixation cross. So really this field started in 1995 with this paper by Bharat Biswal and Jim Hyde as a senior author. And their observation was that if you did task-related fMRI and someone tapped their fingers, then you could get sort of the expected mapping of the motor areas. But their innovation was to look at resting state case where the person is just lying in the scanner, not tapping their fingers. And if you imagine looking at, say, the signal in the left motor cortex, maybe in green here, and you correlate it with all the other signals in the brain, what you end up with is a resting state correlation map that looks very much like the task-related map. Okay, and here I'm plotting here the bold signals from the left and right motor cortices, and you can see they're very highly correlated even in the resting state when the subject is not actively tapping their fingers. So I always like to sort of talk about this paper especially for students, because this paper was, I think, according to Bharat, rejected, I think 3 times at least. It was really violently opposed, and it reminds me of this quote attributed to Schopenhauer: "All truth passes through 3 stages. First, it is ridiculed. Second is violently opposed. Third is accepted as being self-evident." We are now in the 3rd phase for resting state fMRI, where it's accepted as being self-evident, but it was highly ridiculed and violently opposed for many years. And really this discovery languished for probably 10 years before it really started taking hold. Okay, so I think for those of you who are really on the frontiers, it's good to realize that sometimes ideas are just way ahead of their time, and it can take a while to take traction. So by now, as many of you know, resting state fMRI is a very standard process for most neuroimaging. I think sort of the sign that it had arrived was the fact that in 2010, 15 years after the Biswal paper, there was then a sort of a paper in Nature Reviews Neuroscience. So essentially, recounting, at that point, they had gone beyond, the field had gone beyond just motor cortices to map lots of different networks in the brain. And so the tour de force was this paper from the WashU group, from the Human Connectome Project, probably still the best in vivo human mapping of parcellation of the brain using not only resting state, but other methods, but really sort of resting state fMRI to drive this ability to really parcellate the brain in finer detail than had ever been done before. And this paper was published in Nature and, has hundreds of pages of supplementary data. Okay. I think, we can really track sort of probably what started helping the adoption of resting state to this paper by Fox and Rakel in PNAS 2005, about 10 years after Biswall's paper. And this is really sort of outlining this sort of very attractive idea that perhaps the brain is really organized into two anti-correlated networks. And I'll talk a little bit about those networks. The first is what you've probably heard, which is the default mode network. It's centered around the posterior cingulate cortex. And what this map is showing is I'm taking a seed voxel or seed time series in the PCC here, and I'm correlating with all the other signals in the brain. And those areas that are in red are sort of going up and down along with the PCC. And those areas that are going are shown in blue are essentially anti-correlated. So when posterior cingulate cortex has slightly more activity, those areas, which are now sort of attributed to what's called the task positive network or default attention networks, are going in the opposite direction. So this led to sort of this very attractive idea of the brain alternating between sort of monitoring its internal world in the default mode network and sort of monitoring the exterior world through the task positive network, sort of this yin yang of the brain going back and forth. But within 3 or 4 years, this was called into question by Kevin Murphy's paper from the NIH group, where they commented that essentially these findings of this anti-correlation were obtained with something called global signal regression. And this is where global activity starts to enter into the story. And so if you use this method called global signal regression, then you end up with these anti-correlated networks. But if you don't, you don't really typically find these anti-correlated networks. And so the argument of Murphy and others was that this was a mathematical artifact. And so before we get into sort of what global signal regression is, let me back up and tell you what the global signal is and continue on the story there. So the global signal is fairly simple. What I'm showing you here is from a single slice, the raw time series. And these are basically the raw images that you would get off the scanner, scaled around their mean, such that very red means positive bold signal, blue means negative bold signal with respect to the mean. And the global signal is just at any given time point, you just take the average of all the values. And so when the brain is relatively negative, you get a very negative global signal. When the brain has lots of positive values, you get a fairly positive global signal. And in between, and you notice here, even in these single time series images, you sort of see these anti-correlations. And when the brain is in this anti-correlated state, the global signal is close to zero. Okay. So what we'll be talking about is the global signal will simply be this signal, which is the average signal. And then we'll also talk about the global signal amplitude, which is sort of a measure of how large the global signal is. We will use a standard deviation over time of the global signal as our measure of how big the global signal is, okay? Simply because it's typically looks like a fairly random time series, so standard deviation is a really reasonable metric of how large it is. Okay, so what is global signal regression? It's actually fairly standard and basically 2 steps. One is we compute the global signal as the average in the brain, okay, so over all the voxels in the brain. And then we simply do linear regression, and if X sub I is the signal we measure and G is the global signal, we just project out the part that can be explained by the global signal, and we're left with this residual signal here. Okay, so that's all it is. it's literally just one line of code in any pipeline, and yet this one line of code, as you'll see, is very controversial. Okay. Okay, so why is it controversial? Well, what Murphy and others argued was if you look at these histograms of correlation values, and these are correlations using the PCC as seed voxels, then typically you can sort of see they have fairly a positive mean and median, okay? And so this would tend to show very few anti-correlations because most of the correlation values are positive. If you do global signal regression, then you actually mathematically impose this constraint that you have to have essentially equal numbers of positive and negative values, and that they should sum to basically zero. And therefore, even if there were no anti-correlations, you would impose anti-correlations, all right? So the claim is, yeah, that's a very nice picture of the brain, that the brain is anti-correlated, it has anti-correlated networks, but really it's just a mathematical artifact. One way to think about that, Matt, sort of from a geometric point of view, is you can sort of imagine representing each time series as a vector. And in this case, the angle between the vectors is their correlation. And here they'd be, positively correlated, okay? And if you assume that, let's say, take even more abstract view and you imagine a lot of vectors here around a cone and they're all positively correlated, then they would have a positive global signal. That would be the average signal. Now, if you project this component out, they're all going to be, you're going to take out this vertical component, so they're all going to be back on the horizontal axis. And now you can sort of see you've imposed these anti-correlations. Okay, so it's a very simple geometric view. And this is sort of where I got into the global signal. I had a student visiting from China who really wanted to look at the global signal, and I kept telling him, no, that's really not very interesting. But he eventually convinced me, and so we ended up sort of looking more into geometry and going into more detail of what the global signal was. And so then moving forward about 8 years past the Murphy Kevin's original paper, I think this was rather optimistic, sort of towards a consensus regarding global signal regression. I can tell you now there's still no consensus really, but this, even 7 years later, and at the same time, we also published a paper, sort of, updating what we thought was the state of the art then in terms of what the global signal was. And to tell you that, to show you why it's not resolved, there's actually in June, there will be a symposium at the Organization for Human Brain Mapping in Korea. And the topic of the symposium is to regress out or not, an update on fMRI global signal debate. Okay. And so this is still something that the field, either people ignore it or they think about it, or but it really has not been resolved. Okay. So let me tell you a little bit about why people do global signal regression and also sort of alternatives that people are trying. So if you do resting state fMRI and you say place a seed voxel in the PCC, and then you do you try to map the resting state connectivity, in some subjects you'll get quite lucky and you'll find, oh, this looks reasonable. And so this is 5 subjects, and I've picked these obviously so that subjects B, C, D, and E are essentially not usable. Okay, so essentially, if you're a graduate student or a postdoc and you show these to your advisor, they will tell you to go back and redo the analysis, right? There's not much that may be publishable here if you're looking at functional networks. So if you apply, you can apply different things. There's different sort of other approaches you can apply, but by far the most widely used for a long time was global signal regression, for the simple reason that once you do global signal regression, all of a sudden you start to see these networks that appear more, they're first of all more repeatable across subjects, which we'll get into why that might be the case. And also they actually reveal sort of the underlying functional networks that have been reported in literature. And so this is why global signal regression became widely adopted. Oops. So for example, one reason this paper here was showing that if you did global signal regression, you could strengthen your association between resting state functional connectivity and behavior, and essentially, an example of why people are highly motivated to try to remove some of these global signal confounds. This was sort of a Nature Protocols paper reviewing the state of the art of pipelines here, and as you can sort of see, they discuss global signal regression, and it's historically controversial. Okay. And so what has happened is, if you look at what's happened in the field, and if you look at all the pipelines that people are actually using now, there's been this shift from global signal regression to something called CompCore. Okay. CompCore is a very simple idea, which basically says, well, let's look at the signal in the white matter and CSF regions, or else, regions with high temporal variance, and say, though, those are sort of confounds of no interest. And if you do principal components analysis, maybe you can come up with confounds that can remove some of this, these nuisance terms that are not related to the neural activity you're interested in. And this was really driven by, I think, my guess is really driven by this paper from the Susan Whitfield Gabrielli's group, who at the time was at MIT, saying that anti-correlations in resting state networks can exist without global signal regression. And so here you can sort of see with global signal regression, you get these anti-correlated networks, and you apply a CompCore, which is one version of CompCore, you get very similar networks. And I believe this paper, they implemented that in their sort of software FCon, and since then it's been implemented in a wide variety of very widely used pipelines, including fMRI prep, which is sort of out of the Stanford group. And as you can see, CompCore is sort of one of the standard procedures. So I must have to say I have mixed views of this, because it's a student in my group is the one who developed CompCore. Okay, and we developed it way before we were even interested in resting state fMRI. So it's actually not developed for resting state fMRI. It was developed for task-related fMRI, where you actually had some ground truth of what you should and should not take out. And so in task-related fMRI, it's quite easy. You don't want to take out anything related to the task, or you can at least impose that prior. That's a constraint you'd like to achieve. And we were also at the time very stringent in our sort of how we define white matter. And so these are white matter and CSF partial volume maps that we use to identify the regions. And here we use like a 99% partial volume criteria. And our concern has always been, when it's used for resting state fMRI, oftentimes that threshold is reduced to maybe 70% or something, in which case you're getting a lot of gray matter and other things that might have essentially carry the global signal with it. And so this was sort of a comment I made in that review paper, that a potential limitation is that they rely on effective segmentation, and the errors can lead to partial volume effects, in which case you basically are doing sort of maybe not exactly global signal regression, but fairly close. Okay. So I think the sort of the secret is that people are still doing global signal regression. They're just calling it CompCore. But I don't think most people realize that. And just to give you sort of why you might think that might be a problem, here is sort of the global signal, starting off in blue, where it's sort of minimally processed. Then I slowly take out more and more confounds. And you can see going from the red to the cyan, this is where I start taking out white matter and CSF. And you can sort of see that, there is, going from the red to the cyan, there is quite a reduction in the global signal, indicating that the global signal does have, contributions from white matter and CSF. So even if you were strictly in white matter and CSF, you'd still have some global signal. And more likely, for most of the partial volume maps people use, you're probably getting into deep gray matter and regions around the ventricles that are essentially giving you your global signal. Okay. So that's where the controversy lies, and I'll get back later into this talk into some sort of discussion of what might be the next steps in dealing with the controversy. But let me now sort of switch into sort of maybe more the discovery mode, and sort of get into some of the what we think is going on, what is giving rise to this global signal activity. So once again, just like the paper I wrote with the student, this was a different line of research. It wasn't really focused on the global signal. It was more something we were doing in our lab to look at the effects of caffeine on CBF and bowl dynamics, where CBF is cerebral blood flow. And what we noticed is, for example, is this is across thousands of voxels in a single subject. Before caffeine, there's quite a lot of variability, and after caffeine, everything lines up, and you see these interesting oscillations. We also found that caffeine got rid of the initial dip. That those of you who've been fMRI for a while know that this was quite controversial in the late 1990s. whether this dip existed or not. So just as historical context, at the time, the presence of this initial dip was sort of attributed to the initial increase in oxygen metabolism that occurs quite early after the stimulus. So this is in response to a 4-second visual stimulus. And at the time, it was really being espoused by researchers at Minnesota. And there was controversy about whether this initial dip existed at all. And in fact, the folks, I believe, at MGH, Harvard, were saying that no, there's no initial dip, okay? And so I think our sort of tongue-in-cheek conclusion after writing this paper is probably the people in Minnesota didn't drink much coffee, so they could see the initial dip, and those in Boston drank a lot of coffee, so they never saw the initial dip. And essentially, that was the reason for this controversy. Just as an aside, taking caffeine, in this case, 200 milligrams of caffeine, reduces blood flow by about 30%. And so we were able to use this model to sort of, look at the biomechanics of, how the arterioles constrict and how changing the blood flow would affect their constriction, able to sort of model quite nicely sort of variations in the dynamics similar to this, based on looking at sort of the bioengineering aspects of the vasculature. So this was sort of the line of research we were on. We then decided, because resting state was becoming more popular, to look at resting state. And sort of to our surprise, we found that if this was the resting state map before caffeine, the resting state map in motor cortex after caffeine essentially was largely diminished, okay? And this actually sort of went against our thinking, because at the time it was thought that you could use caffeine to increase the bold response. It would be faster. It would also increase in amplitude in some cases. And there was, even some work from the group at Northwestern looking at its effect on metabolism. So all these things pointed to the fact that this effect should have really gone the other way, that we would hypothesize that caffeine would improve these connectivity maps. And so we were quite surprised by the fact that it sort of eliminated this functional connectivity to a large extent. And so at the time, really, at that point, I'd only done BOLD and CBF measurements. But as much modeling as we could do, and sort of, we realized that we really couldn't answer this question with just BOLD or CBF alone, that we really had to get into multimodal imaging. So this sort of represents, my own sort of entry into multimodal imaging, which for a first dive into multimodal imaging was sort of, in retrospect, I'm surprised I did it, but it was sort of like this very painful experiment where we only ended up doing 10 subjects, but each subject did 4 scans. So they did 2 scans or actually 4 scans. Yeah, they actually did 8 scans total. So basically, they did EEG fMRI in the scanner, caffeine or placebo, pre and post caffeine. Okay, so that's already 4 scans. And then we took all these subjects and took them into the MEG and did simultaneous EEG, MEG, caffeine or placebo, plus eyes open or eyes closed. So everything was all these variations that we could think of, and did that also pre and post. And so that's the data that I'm going to spend the next 5 or 6 slides talking about. And I'm still surprised we got through this study, and it was quite a painful study. And just, I think, not going to go into too much detail, but essentially, the main reason we used MEG and EEG is because we really wanted to sort of decouple sort of the electrical, sort of neuroelectrical sources from the vascular confounds. Okay, so in addition to having lower temporal resolution than MEG and EEG, one of the things that makes functional fMRI a bit difficult to interpret sometimes is the fact that as blood flow changes and metabolism changes and blood volume changes, that can affect your BOLD signal in ways that we understand somewhat, but not completely. Okay, so I'm just going to show you, because I'm going to show you maps of, I've shown you some sort of connectivity maps, but the other way we look at functional connectivity is with functional connectivity matrices, and the concept is fairly straightforward. We take sort of pairs of brain regions and we correlate the two, and then we enter that correlation value into a matrix. And we do that over and over all the different pairs of regions of interest that we might care about. And we would end up with a functional correlation map like this, a matrix like this, where the red indicates positive correlation and the blue indicates negative correlation. Okay. So what we did was, the first thing we did was we sort of verified that we took, the previous study had really been focused on motor cortex. Here we showed that this reduction in connectivity was actually whole brain. So this is caffeine pre and post, and you can sort of see a reduction in the connectivity values. And in control, we don't really see too much difference. And this is the matrix view. This is if we just pick a PCC map, and you can sort of see here, large, very positive correlations, and here a diminishment in the positive correlations, and you're starting to see these sort of negative anti-correlations. And so, sort of tongue-in-cheek, we dubbed this natural global signal regression. Essentially, if you look at the data without global signal regression before caffeine, and you apply global signal regression, you get these anti-correlated networks. And if you have the subjects take caffeine and don't do global regression, you also start seeing these anti-correlated networks appear. Now, not exactly the same, but this was sort of a clue as to what was going on. Okay. And in fact, what we found is, if you looked at the global signal amplitude, we found that the caffeine led to a reduction in that. Okay. So if you do the same thing with MEG, we found, so one of the things that we found is, here's the bold, and you're sort of seeing that this is the one showing reduction with caffeine. And similar to first order, you can see qualitatively in the single subject, this is where you're seeing, with the caffeine, you're seeing a reduction in MEG connectivity. Here we're using sort of source space analysis, using beamformer analysis to look at connectivity between different regions with the MEG. And similar in the here we're looking at MEG wideband, caffeine versus control, eyes closed versus eyes open, and here we see a fairly strong reduction in the eyes closed condition, not so much, still reduction in the eyes open, but not so much. And this also mirrors what we're seeing with the bold, much stronger reduction in eyes closed versus eyes open, and control, some indication, but not really as global. And then sort of the more tortuous experiment we did, and I think anyone who's done simultaneous EEG fMRI knows it's not something that you want to do unless you really need to do it. But we felt we really need to do it. This is what the data looks like on the EEG due to the gradient switching. Luckily, many very smart people have developed methods for cleaning up the data such that by the end of the day, you are able to get reasonable looking data. Now, what do you do with EEG? So there's lots of things you can do with EEG. What we ended up doing was eventually sort of focusing on sort of EEG as a measure of arousal or vigilance. And this is a table from one of our review papers showing that there's lots of different ways of measuring vigilance. We picked something that's fairly simple, which is we dubbed EEG vigilance, but in the literature, it's also known as the alpha slow wave index, as essentially the ratio of alpha power over delta and theta power. Okay. And as so this is here, this would be a subject with a relatively high state of vigilance, and the alpha power is higher than the delta and theta powers. Okay. Now, sometimes, especially, for example, even the reviewers ask us about this because there is this sort of, everyone knows about the Berger effect, and when you close your eyes, alpha goes up, right? But it turned out that's a fairly transient effect, and so what we're talking about is more steady state effect in terms of if your eyes are open or your eyes are closed, what's happening over a 5 or 10 minute period. Okay. So what we found was very interesting, actually, and it was actually once again one of these things that we didn't anticipate, which was initially we only had 10 subjects, so we were actually quite disappointed that some subjects actually showed almost no effect of caffeine. Okay, so here in this subject, you can sort of see their EEG spectra are fairly similar before and after caffeine, and their resting state bold maps are also fairly similar. But other subjects showed quite a large effect of caffeine. And so, for example, in this subject, before caffeine, they're showing relatively low vigilance, and you can sort of see lots of positive correlations. And after caffeine, they're showing relatively lower vigilance or higher vigilance and more anti-correlations. And so it turned out that, this intersubjective variability obviously affects your ability to, show a group effect, right? But it actually is a nice natural variation that allows you to show other relationships. And so we were able to use that relationship. So we could look at the change in vigilance per subject versus the change in global signal amplitude. And you can sort of see that the more the vigilance changes, the more the global signal changes. Okay. And this is across the subjects. And I would have to say this is about as good as it gets in science, right? In terms of, on human subjects with such a difficult experiment. Okay. So we did the same thing with eyes open versus eyes closed, and we saw the same sort of intersubject variability where some subjects, eyes open, eyes closed, has almost no effect on their EEG spectra, and others show a large effect. And so here, the eyes open really does increase their vigilance level. And interestingly enough, if we plot the eyes open data and the caffeine data on the same axis with delta global signal amplitude versus delta vigilance, we see essentially, they're sort of showing the same effect. So to first order, didn't really matter how you change the vigilance. It was really just the relationship between vigilance changes and the global signal amplitude appeared to be similar. And then also the variations across the sample actually gave us a nice natural experiment in the sense that some subjects show high vigilance and anti-correlations. Other subjects showed very low vigilance, or sort of medium vigilance, and sort of medium levels of global signal amplitude, and other subjects showed other values. And so we started to see this over the sample. We can sort of see this sort of relationship between global signal amplitude and vigilance. So as people become more vigilant, their global signal amplitude goes lower, and as they become less vigilant, there's much more global activity. And so this is basically this overall effect you'll see holds up across other studies done by other groups that I'll talk about in a bit. Okay, there. And so sort of one of the nice examples of that is sort of in this study a few years ago in Nature Communications, where they're looking at sort of bold standard deviation, which is sort of similar to basically global signal amplitude, and you're sort of seeing here where it's low, okay? So at this point and this point where it's low, you sort of see less detection errors in a visual detection task, okay? And this is occurring sort of at the times of the day where the light is sort of, between, it's like either around right around dawn or right around dusk. And so the point of this paper is that perhaps the brain has evolved to have lower global signal amplitude in these areas, or where you need higher arousal. And so essentially at these points where you actually need to be able to detect things where the light is sort of funny, that the global signal amplitude goes down, and your ability to detect visual objects increases. So overall, we have this picture where as vigilance is higher, we expect more anti-correlations, more global activity, and similarly, when vigilance is low, we'll see higher global activity and much more global functional connectivity. And one of the things that as people are looking at their data, global signal actually is becoming adopted more almost as its own metrics, not just as a confound. And so, for example, in this PNAS paper from the Yale group, they're showing global signal is higher in schizophrenics versus human controls or healthy controls. And their hypothesis was that essentially there were some very intricate sort of neurocomputing sort of modeling of this that would lead to this result, but there's also much more parsimonious interpretation, which is it's sort of known that in schizophrenic populations, vigilance levels tend to be lower, and so therefore you would expect a higher global signal. Okay, so I'm going to go back and sort of revisit this slide here and sort of start taking apart like what might be going on with the global signal and what's, what might it tell us about what's going on in the brain and how we should deal with it moving forward. So some of you may know who Yogi Berra is. Hopefully at least one or two people know who Yogi Berra is. He's a New York Yankees baseball player, former coach, and some call him American philosopher, known for Yogi Berrisms, like you can observe a lot by watching. Okay. And I think this is really important in this day and age of big data, where a lot of our trainees, the newer people to the field, have maybe even never collected their own data or even looked at raw data, and it's all these big databases and there's all machine learning that we can do. But I like to show this picture because this is showing you the sort of the raw data, or somewhat processed data, and you can sort of see here that you're looking at what the bull signal is doing, the spatial temporal patterns over time. And as I pointed out before, just by looking at the data, you can say, well, yeah, it looks like there are places where the brain signals are anti-correlated, even without doing any analysis. Okay. So Xiao Lu and Jeff from Jeff Dunn's group at the time at the NIH sort of made a similar observation, which was that if you looked at a brain region such as the PCC, and you just sort of said, well, let me look at the time points where those signals are higher, okay? And they call those co-activation patterns. If you did an average of all those time points where that signal was high, you would end up with a map like this. And this is just a time frame average, okay? So it's no correlation, it's nothing, it's just averaging data. And that looks almost exactly like what you get with a correlation map, okay? We can play the same game with the global signal amplitude. So in this case, the blue is the global signal amplitude. And let's say we censor out, we say, okay, let's look at all the points where the global signal is not very high. And so in this case, let's just keep the points, say here, in blue or red, where the global signal amplitude is relatively low or below some threshold. And you notice here is the PCC map that we obtain with global signal regression. And here's what we do is we just retain these time points here where the global signal is not very high, and you sort of see we get something very similar, okay? So the anti-correlation pattern that's seen in this map is already in the time points that we would retain if we just got rid of all those times where the global signal was high. And it turns out that these global signal, if you start looking at this picture of saying, well, it's not like we wanted, when we acquire data, it's actually important to note that there are actually transient events that are driving a lot of what we're seeing. And that if we're not aware that there are these transient events, then our analysis of the data is going to be quite different. This is also from Xiao Lu, showing that here he's looking at what's going on at these points where the global signal is high. Okay, and here he has EEG measurements, and he's able to sort of see this sort of basically average sort of changes in gamma increase, alpha beta reduction, and then post transient event delta waves. Okay, and in this study, he was able to show that most likely what's happening is sort of before the global signal, the signal gets high in the cortical areas, which has been increased in the global signal, you're already seeing sort of changes that precede that in the thalamic and brainstem areas, okay? And areas related to arousal transients, okay? So if you sort of take this game a little further, you can imagine if we have all the data points, this is just sort of summarizing some data points here where we have these points that are anti-correlated. We also have time points that are very positive or very negative and very positive. And then we do the correlation, we end up with this map that we talked about before. It turns out that global signal regression largely preserves those time points where these anti-correlations exist, and they largely downweight. So going from here to here, there's a large downweighting of that signal such that these points become downweighted. And in the extreme, we could even censor them. So they become zero. And so you end up with global signal regression and censoring offering very similar views of what's going on. And so what we've done is we've retained the time points that have low global signal magnitude where anti-correlations already exist. And we've censored out those points that have either high vigilance or low vigilance, or the raw images have a lot of spatial homogeneity. And in essence, global signal regression is attenuating the effects of vigilance variations in a resting state time series. And so after the censoring, the anti-correlations do exist in the periods where the global signal is low. And for those time periods, there's really no mathematical artifact, right? All we've done is just pick those times and looked at the correlation in those times, or even just averaged it. And so I do think that this sort of says that, yes, there is this mathematical artifact due to global signal regression, but perhaps in the future, what we need to do is more of a sort of a time multiplexing view or a time division view of our data to get a better sense of what's going on. Censoring obviously is a very blunt approach, but perhaps in the future there may be more sophisticated approaches that can sort of parse the data into different spatial temporal cohorts. So getting to that, what might be going on, essentially we're going to get into sort of spatial temporal patterns now, which is sort of traveling waves or patterns that move through the brain. Okay, and so this is influenced by the work out of Sheila Kellholt's lab at Georgia Tech and Emory, where they show that if you look at the brain data, there are sort of these repeating patterns. So here I'm showing you 6 slices over about 30 seconds, and you sort of see these patterns of activity move across the brain. Okay, and these tend to be repeating motifs. And in fact, if you can identify these motifs and sort of estimate where they might occur, and you average those motifs, you end up with a signal that looks very much like the global signal. So the blue is the global signal of the original data, and the dotted lines are what you would get if you just assume that the brain data was just made up of these spatial temporal motifs moving through the data. Okay. Since then, there's been a lot of work showing that probably what's going on is a lot of the global activity is dominated by these sort of transient traveling waves moving through the brain. This is work also from Chalou's group showing that looking at the sort of the principal gradient moving from either the default mode network from motor areas to the default mode network, or from default mode network to sensory motor areas, that there are both bottom up and top down traveling waves. And the ratio of those two tends to depend on a level of arousal. So for example, when there's a lot of drowsiness, okay, there tends to be much less of these top down waves than bottom up. And they become a little more equal when there's sort of high levels of alertness. There's also been work from the WashU group and also from UCLA group showing similar analyses, suggesting that really the way we start looking at the brain is not, using correlation analysis or other approaches, but really looking at these sort of traveling waves of activity going through the brain. And this work from Taylor Bolt, essentially what they're seeing is that the global signal is really related to this sort of this sort of traveling wave that has this sort of specific pattern moving through the brain. So you can sort of fairly negative here and then fairly positive here. Okay. And so the idea is, current analyses don't really take into account these spatial temporal patterns, and maybe what we need to do is the methods need to sort of move in that direction. Now, one thing to keep in mind when doing any resting state, especially in humans, is this is some nice work from the Taguchi and Helmut Lauf's group, showing the probability of being awake over time in a resting state fMRI scan. And as you can sort of see that, like this is about 10 minutes, 5 minutes, even 5 minutes in the scan, the probability of your subjects being awake is at most a half, okay, or here 0.7, but dropping down to half by 10 minutes, okay? And so a lot of the analysis to date has been done on things like the HCP data set with 15-minute scans. And so what we're really seeing is a lot of drowsiness effects, okay? And you can even sort of see that in a study aiming at drowsiness effects, where we're looking at microsleep things, you can sort of see that you're seeing these huge global effects at sleep onset, at wake onset, sleep plateaus, post-wake undershoots. There's just a lot of stuff going on as people are coming in and out of wakefulness. Okay. So I think that's something that in general that, it is important to keep in mind, especially for those who are looking at this type of data. Now, one question is this only a problem with do these transient arousal events only occur in humans that are sort of falling asleep? This is some data using sort of the visual coding data from the visual coding data set from the Allen brain map, showing that actually, and this is in, sort of awake mice, all, obviously going through different arousal states, but, they're still running on a ball, so they're not completely asleep, and showing similar effects that essentially you do see these sort of propagations of transient arousals that are linked to sort of, in this case, pupil diameter, okay? And in fact, they went on to look at the data even some more, and these events occur not just during resting state, but also during passive viewing of scenes. And what they found was that if you sort of looked at what happened before and after these arousal transients, after the arousal transient, there's sort of an increase in decoding ability. And before the transient, there's much more sort of hippocampal ripples, okay, indicating that even within the brain may be set up, around these transient events to go between, you're sort of recapitulating, sort of consolidating memory even in real time, and then going to encoding and potentially this, in a way, almost reviving this picture that we started off with of anti-correlations, but just at a different time scale here. Okay. That perhaps the brain has to have this ability to go between the external and internal world, and maybe it's even happening at faster time scales than we had thought. And in fact, I think, sort of to sort of close, I think what we're finding is that this is from the very influential paper with Stringer and others from the UCL group. Also, this is neuropixel recordings, not from the Allen Brain Atlas, it's a different data set, but just showing that when we look at the brain, there's so much going on. And this was sort of a study where they essentially looked at the neural activity in the brain, and then they also looked at what the mouse's face was doing, okay? And just by looking at the principal components of, the face structure, they're able to predict sort of this sort of spontaneous fidgeting of the brain, essentially predict what the wide scale neural activity was doing, okay? And I think this actually has implications for those of us doing human neuroimaging. This is a preprint from Bolt and others, again, showing that, if you look at global fluctuations, it turns out that if you look at almost any other metric or physiological metric, what they'll find is everything is correlated with everything else, okay? Essentially, if you look at global signal amplitude, which in this case is their first functional component, it's related to breathing, it's related to your cardiac sort of pulse wave pressure, it's related to your heart rate, it's related to pupil dilation, it's even related to cerebrospinal fluid, lymphatic flow. It turns out that the global signal can actually be used as a measure of, how well the lymphatic system is working and disposing of waste from the system. And obviously there's relationship to EEG power. Okay. So, and then this another paper from Gu and others showing you as these transients occur and global signal activity is changing, there's just so many things happening at the same time. So salience network deactivation, developmental network activation, increasing global signal here, respiratory changes, heart rate reductions, alpha power reductions, CSF inflow. So essentially this picture of looking at the brain in isolation, I think the other thing that we need to start appreciating is, the brain obviously evolved to sort of control the body. And if we just look at the brain in isolation, it may not be it's certainly not giving us the complete picture of what's going on. And I think this is my last slide in terms of content. And I think, we're definitely in a sort of a golden age of, where there's so much data available now, okay, driven by, lots of open source projects and large scale consortiums. And we're also in the age where, obviously there's deep learning, large language models, lots of pipelines that are available. And I think this is great because I think there's lots of discoveries that can be made, but a lot of these are just finding different correlations between different aspects. And the more you look at the data, the more correlations you will find. And the more variables and the more data you have, then obviously you can find many more. This is correlated to that, and this is correlated to that. And so the question is, what do we do, and how do we get out of this forest, where it's like the blind men touching different parts of the elephant, and what is really, what is really at the center. So I don't really have the answer for that. Hopefully, the audience can help with that. But I would just say one aspect I do think we need to keep in mind is that although we are imaging the brain, certainly we need to sort of start taking into account the effect that when we look at the brain, if we go to that elephant analogy, we're really just studying maybe the trunk, right? And we're ignoring the fact that it's really hooked up to lots of things that are, the brain is both controlling, but also influenced by that. And the circularity of things is, I think, what leads to all these correlations. And so whether or not we can find out our way out of this forest of correlations, I think is an open question. With that, I'd like to thank you for your attention. Thank all the great students and trainees who helped with this work, and the Whitaker Foundation and NIH for funding this work. And with that, I'm happy to take any questions. Thank you.
Great. Thank you so much, Tom. That was a fantastic talk. We can open up. I have a few questions, but why don't we open up to the floor? So why don't you put your hand up if you have a question? Hopefully I will see you. Antonio, why don't you go ahead?
Hi, thank you for the very interesting talk. It's a big picture question. To me, part of the problem when I was listening to you about the controversy and the different angles with the global signal is that we still model largely one voxel at a time. And then we understand that there are relationships between the things and potential confounds, and somehow we use global statistics of one form or another, a statistical methods to take them out. But at the end of the day, isn't the only way to get at that is to do the statistics of the dynamics of these networks all at once, and then figure out how they are, what is endogenously, whatever common component on these signals are, and whether it's not. But if we don't do that, we're not going to crack the problem. Question one. And question two, has anybody been trying to attack this question by modeling the entire network at once?
Yeah, I think
we had to downsample not to 50,000 voxels, but now we can do parcellations of a couple of thousand areas, and we do the dynamic modeling of those things.
Yeah, no, I think that's exactly the right. I think those are certainly the questions, and I think certainly the getting a better sense of how to model spatial temporal processes is part of the answer. There has been, large scale modeling. Gustav Decko's group, has really sort of pioneered a lot of that. I think the concern is that, I think things are moving in the right direction, but to do the modeling correctly, I mean, they're typically modeling, is biased in the sense it's trying to reproduce certain phenomena that have been observed, right? And so I do think that whether the model is correct or not is always biased behind what it's trying to show, and so, but I do think that is the right answer. But it's actually going to be quite complicated in the sense that to actually have enough data to inform the model is going to be a lot of data. I think once you want to measure all the neural activity and certainly what the Brain Initiative project is sort of aiming at, sort of measuring more and more things with neuropixels and things like that. But I do also think that part of the multimodal imaging is going to be measuring also what are sort of the physiological behavioral things that the body is doing. There's lots of twitching and fidgeting and just stuff going on all the time, and that's turning out to be a large component of, especially in the mouse data, they're finding that all this stuff is a large component of actually what's going on in the data. And without knowing that, you might interpret it as being something else.
Thank you again.
Yeah.
I think John is calling on me, but I'm not entirely sure because he's muted. Can you hear me all right?
Sorry, yes, I was calling you. Apologies.
Yeah. So thank you for a great talk again. I was wondering, so yes, a lot is happening in the brain, and it would be lovely if we could look at all the things at once and have a better sense of modeling them spatiotemporally. And this was all in the context of resting state data. All of this stuff is also happening when a lot of us are doing task stuff too. So how do we think of how to model task data if we were to take all of these things into account?
Yeah, that's a great question. I left it out of this talk, but actually the global signal regression actually happened many years ago, even before resting state. And it was shown that if you did global signal regression, it would totally change your interpretation of your task-related data for many of the same mathematical reasons that it affects resting state data. So I think, people have been finding and probably, there is a lot of appreciation that the background fluctuations, what state you're in, has an effect on, your decision making or your, response times. And so I do think that people are aware of that fact. I think the hope is that, at least for task related, there is the hope that because you know what you're looking for, if you average over enough subjects or enough scans where that effect averages out, then, maybe you can ignore it to some extent. Or you can track it. You can either use it and see, or you can just hope it averages out.
Yeah, it sounds very hopeful, but yeah, okay, I'll keep holding on to that hope. Thank you.
Okay.
Yeah, so actually, Tom, I wanted to follow up on that question. So, because it seems like a lot of the task fMRI stuff, at least that we do at Caltech, and I think that a lot of people do, is like doing things like inducing emotions or have people kind of vary the value, the significance of things, and obviously that's probably going to impact disproportionately on vigilance, right? So you're going to be more vigilant, more aroused in certain conditions than in others. And so I wondered, how nasty that could get in terms of the types of issues that you're describing.
Yeah, I mean, I think the good thing is if you have something that is keeping the subject in a relatively vigilant state, then these large global fluctuations are diminished to a large extent. So that helps you. And so then the sources of variability are if it's a very demanding task and they're losing vigilance, and that could be something to look at if you're seeing some effect that doesn't really make sense.
Right. And another thought was, to what extent then, given what the results you described where, people just, which we all knew, people get tired and they start even when they're doing task fMRI, and probably even way more so in resting state, over orders of minutes they start to get tired. I mean, is that something we just need to better control for? And I'm just thinking, for example, one could imagine some kind of very constant task, right? That is where they're constantly having to do a task and you're collecting a kind of a type of resting state, but it's not resting, but where at least you're controlling the state that they're in, right? In other words, is what I'm trying to think about. Imagine having to do some constant keep looking at a moving dot or something, and I wonder whether that would be a better way of trying to control for these vigilance effects. I don't know what your thought is.
Yeah, I think anything that sort of requires a lot of attention over time will sort of cause, it's very difficult, right? I mean, I think the hardest task that I've had to do in the scanner is, eyes fixation for 10 minutes. I mean, by minute 9, your brain's just going crazy. It's like, it's a really hard task to do. And it probably would be easier if there was something to do. I know what the consortium, such as the HCP, the original HCP used 15-minute resting state scans. And then when they went to the lifespan HCP project, they purposely reduced that to about 6 minutes. And what they found was they got much better results because people could reset, and they take a break and then reset, and there was much less motion, and they're sort of better able to control for these confounds. So I do think where possible, certainly shorter scans are better. 6 or 7 minutes is good, but these long scans, then you are getting into realms where the brain will start doing something after 5 or 6 minutes, that may not be what you wanted to do.
Zeynep, another question?
Yes, I'm back. So I understand your expertise is more in the human resting state data. You did mention some animal model work, but and I'm sorry if I just spaced out and didn't catch this properly. But I was just trying to understand what is the state of understanding in terms of how these global signals relate to actual electrical activity, like single neuron activity, population level activity. Is there a good sense of that, or where would I be looking at, or what should I be looking at to get the sense of that?
Yeah, my understanding, as you said, it's not my area of expertise, but that visual coding data from the Allen brain map is sort of spike activity, and I believe what they did was they sort of averaged mean activity and looked at sort of peaks and troughs in the global mean activity, filtered around 0.5 hertz, and so basically use that as the indicator, and essentially we're looking at those neurons that sort of fired before and after those troughs and peaks in the global signal. And so they essentially applied techniques from human fMRI to the animal data. And so I think, obviously, one of the things with multimodal imaging that I sort of glossed over is that, how do you compare things from different timescales, right? Initially, when we looked at the MEG data and EG data, we're thinking, okay, there's these, tens of millisecond timescales. Should we be doing correlations at those timescales? What we ended up doing is sort of, doing what a lot of people have been doing, is basically, either take the Hilbert transform or, somehow compute the power of those fluctuations. And that tends to be the power of those fluctuations over, much slower timescales tends to be what's more correlated to the fMRI signal. And, but I think it's something to keep in mind that there are different timescales going on. And I think that the global signal timescales that we see in humans, that's one time scale, but I think the animal data are showing that there's also other time scales, that there are global traveling waves, and those also will have. So I think there's going to be sort of this nested thing of different spatial and temporal scales that are all going at the same time. And I'm sure evolution has figured out all those ways of keeping things running.
Right. Okay. Thank you.
So let me ask another question. So I was also kind of struck by, the caffeine data and obviously the effects that it has, which seems to be quite substantial on the data that we collect. And obviously, it speaks to the idea that there's probably going to be huge differences across, individuals or across studies in terms of the extent to which those global effects are impacting on the data, depending on, as you mentioned, have you had a cup of coffee that morning? Did you have a busy, did you go party last night, right? All these things that normally when we're doing studies, we normally wouldn't even control for or even think of controlling for. So I just wondered to what extent you really think that's something that we're just going to have to take more seriously to try and understand, what state is the person in before they go into the scanner, as well as, of course, what state they're in when they're in the scanner.
Yeah, I think it depends a lot about, on your sample size, right? And whether these effects will average out and what you're looking at. Typically, the recommendation I gave whenever people ask me is try to just measure people in their sort of steady state. So, if they have a cup of coffee in the morning, try to measure them not right after the cup of coffee, but, sort of mid-morning, between their caffeine, doses. And because otherwise, if you ask them to abstain, then you're measuring them with withdrawal, which will cause their blood flow levels to go up, and that's going to have other effects.
Mike.
Yeah, actually following on that point about the caffeine, and I've actually seen similar results for like the loss of bulk contrast when you take alcohol as well. So there's a fair number of things you could potentially take in everyday life that might affect the bulge response. This is the first time you mentioned the kind of dawn-dusk effect. And that seems to be a fairly strong effect on global signal. Is that something you could potentially exploit by doing fMRI studies at dawn and dusk when apparently we're like slightly more attentive? Or is it something, again, that you need to control for? Don't do it too early in the morning, don't do it too late in the afternoon, kind of like keeping away from breakfast and caffeine and things.
Yeah, I mean, I think that's a good point. I mean, I think, you know, one certainly could, we haven't really in my lab looked at that circadian effect too much, but I think it's certainly, once again, another source of variability that, you know, whether you average it out or try to take advantage of it, I think depends on the logistics of the experiment, I would think. Yeah.
Sure. Okay, thanks.
Sure.
Great. So if there are no more questions, I think we should thank Tom again for a very thought-provoking talk. So thank you very much for spending your morning with us. Hopefully you didn't miss your coffee while doing so, and we look forward to keeping in touch and hearing about your research. And
Great. Thank you so much for having me. Okay.
Thanks very
Bob.
much.
Thank you. Okay. Enjoy
Take
your
care.
weekend.
Bye,
Okay.
everybody.
Bye. Bye-bye.